So this is a continuation of the previous slide. As you can see, when we're trying to link two monomers, that's two groups of atoms, right? Two groups of biomolecules that we want to make into one macromolecule. Macro meaning large, right? So macromolecule. So when we're trying to make a macromolecule, we're going to take two monomers and link them together. Well, how do we link them together, right? What's that process called? It's either going to be called condensation reaction or it's going to be called dehydration synthesis. Again, something we just covered on the previous slide. Now, dehydration synthesis and condensation, all that is the removal of water and the recombining of the bonds that used to hold, in this case, OH and this H together to the molecule. You take those away, that leaves these electrons available for bonding. So then you get a bond formed. This is the bond that's formed right here. That's called the peptide bond. And because there's two amino acids, they call this thing a dipeptide. So of course you can have a bunch of these together, hundreds together, right? Hundreds long, as we saw with hemoglobin. See hundreds of these together, and the process is continuous. It's called dehydration synthesis to put them together. To do the opposite, to break them apart, is called hydrolysis. So that's the breaking up. Hydro meaning water, because you're adding water. So you took water out to put them together. You're putting water in to take them apart. The H goes and takes over the bond here and at the one, and the OH takes over at the second, and you get two amino acids again. So you break up the proteins into amino acids, or you can put them together into a protein, or break the polypeptide up to form amino acids, or you can put them together to form a polypeptide, however you want to look at it. The bottom line is that these are the two processes that are involved. What are those processes? Condensation or dehydration synthesis, and the other is hydrolysis. They're moving in two different directions. And here it is again. Hydrolysis is breaking up, where condensation is building, right? So that's really what you need to consider. And of course here, remember that it's called the peptide bond that's holding them together, which is why we call a bunch of amino acids put together polypeptide, right? So here, we'll discuss the various roles of amino acids. You can go back on the previous pages and write those down. We talked about them already. Amino acid structures can form, or polypeptides. Some polypeptides are proteins in themselves. Others are combined to form a protein. And proteins can do anything from function as an enzyme, and we'll talk about what enzymes do later. They can function as structural, so your skin, your hair, we've talked about that a lot. Proteins can form functions that can act as color absorbers. They can absorb light. So they can act as pigments, and we looked at that in our lab. So there's a lot of different things that proteins can do. They really are the workhorse of a living system. So they do a lot of the work that a living system needs to do. All right. They are the bricks, if you will, where your system is the house. So describe what makes up 20 amino acids found in proteins unique. Now, again, I want to mention that I made a mistake in the previous video. I said 21 is 20 amino acids, and an amino acid has this R group that sits out, and you were tested on that in your previous quiz. You have four basic parts to an amino acid that surrounds a central carbon. You have a carbon in the center. You have an R group. You have an amine group, which is an H2, you have a C double bond O, OH, which sticks off to the side. You can see it up at the top here on this page. You can see it on the previous pages, what have you, and then you have this H group. So the carboxyl group, the amine group, and this R group is what makes it different. Each one, this is what's different in each one of the 20 different amino acids. If the amino acid is acidic, or if it's basic, or if it's what have you, it's this group that's gonna make the difference. It's the R group, because each R group is different. It's chemically different, it's structurally different, etc. Describe the process that determines the sequence of amino acids in a polypeptide chain. You don't have to worry about that yet. We haven't covered it. We'll come back to that. Explain how the chemistry of amino acids enables them to act as buffers. Again, let's not worry about that. You know what? I think we did cover in general, even though the details we haven't covered. Make sure you know that it's DNA, right? That codes for RNA, that leads to the primary structure. All right, we'll see that. We'll see that primary structure again. We saw it in the case study. We'll see it again in the next couple slides. Giving examples, explain what is meant by an essential amino acid. And again, the essential amino acids, we talked about it. These amino acids are amino acids that cannot be made by your body. They have to be eaten. The medical students quizzed you on this, those of you that went on the field trip. Describe the process by which amino acids are joined together and broken down. That you should know again. We just covered it again. I've covered it several times. It's dehydration synthesis. Or condensation reaction to build, right? And the other is hydrolysis, hydrolysis, lysis meaning breakdown, hydro meaning water. So using water to break down. All right, good luck.